I'd like to welcome people online and in person to E443 543, which has got a number of names, computational biology modeling methods, which is pretty generic. What we're going to be talking about are what I like to call virtual tissues, which are agent-based models of multicellular organization. And Paul Macklin's assistant's actually teaching a course on a similar topic this semester, focusing on cancer applications. This is going to be maybe a little bit broader, talking about the general modeling principles. So in a sense, the courses, I think, are probably pretty complementary. As always, you're on the Zoom, so you know that it's being Zoomed, and it's also recorded and made available on YouTube, so that if you want to watch anything again, it's available to you. I need to remind you of that. So today is very much a getting started lecture. We'll do a little bit of computing, but not a lot. And for some people, this is going to be the nth time they've heard this lecture, but that's okay. So the topic of the course is going to be the structural and functional organization of organisms, specifically looking at how multicellular organization works, how it's built, how it's maintained, and how it fails. And we're going to be exploring how to build computer simulations, predictive models of that complexity to understand how organisms are made, persist, and fail. Those are fairly complex topics, and the amount we're going to be able to do in a semester is going to be relatively limited. But the hope is that by the end of the semester, you'll have the basics that would allow you to go forward and apply these ideas in your own areas of interest. Today, we're going to be talking a little bit about course logistics. Then I'll talk a little bit about why this might be useful to people. We'll introduce the methodology of the software. We'll do a couple of exercises using the software today. In general, the reason it's organized the way it is sort of 1 3-hour block in the afternoon, is that the course is sort of a hybrid between a lecture course and a lab course, a computational lab course. So there'll be some lecture, then will be some exercises in class, some lectures. I'll go over some examples of simulations that have been written using this kind of software. There are a lot, and we certainly won't be able to touch on all of them, but I hope there'll be some that are of interest to you. I'd like to hear a little bit from you about your background and what you're interested in. I'm not going to push projects right away in the semester, but this is a project-based course, and it's helpful to start thinking about what might be a nice project for people from the beginning. Don't feel shy if you don't have any ideas. It's always possible to adjust later. And then we'll talk a little bit about the computer simulations and how they work. So the course is being taught by me. We meet in the afternoons, late afternoons here, and we'll have this online option for people as well. There may be weeks when I'm traveling or I'm sick or somebody else is sick when we have to do it online, but I'll try to be here as much as possible. We don't have a formal AI, but Hayden Fennell is available to help. Michael Getz is a postdoc in my group who knows Computational quite well. Maciek Swat is the lead developer of the software, and he's usually very accommodating if there are problems. We'll use primarily, I'll try to put things up on Canvas, but normally the online resources will be mostly in the Google Drive. I hope people were able to get to the Google Drive. I'm not a big believer in tests, so there are no tests. There will be weekly homeworks, maybe occasional weeks when we don't have one, but mostly weeks there'll be short homeworks. You'll be doing a project, and you'll be doing an oral presentation of the project. Participation in class counts a little bit. I'll ask people to do a critique of a classmate's project, assuming that things work out that way. Give you some credit for that. And sometimes if people feel like doing a little video explaining their project, I'll get extra credit for that. And some people may need to have a little bit of extra background in Python. The software we use is scripted in Python. You don't need a lot of Python, but if you haven't done any programming, you may need a little bit of extra background. And so if you want to complete the Rosalind tutorials online, for example, I'll give some extra credit for that. This semester, I'm going to be a little bit stricter than usual about late homework. Sometimes it's easy to get behind on these homeworks, and the homeworks exist not for the purpose of grading, but for the purpose of helping people learn the material. And so I think if you can't get them done within a reasonable amount of time, then it's better just to say okay and move on to the next homework problem. Don't worry too much about finishing everything. The assumption is that the main point of the homeworks is to help you learn enough to be able to do the project. Before we move on, any questions about the structural organization of the course? Does that make sense for people? I had originally a longer schedule. In the end, life tends to happen, and so the schedules change. Today, we'll have a very broad brush introduction. Next week, we will start talking about how to create in the software virtual cells and have them have volumes and stick to each other and move. It's not terribly exciting, but until you have cells as agents, you can't do anything with them. And then the following week, we'll talk about how to control cell growth and division. Once we have cell growth, division, and movement and adhesion, we can do quite a bit biologically. And then the following week, we'll start talking about chemical fields, how to control secretion, absorption, cell response to chemical fields. And then beyond that, we'll talk about how cells move in response to chemical signals. We'll begin to build a little avascular tumor spheroid simulation. Later in the class, we'll talk about intracellular control of signaling and gene regulation. And then I hope we'll get into some more advanced topics this semester: compartmental cells, epithelial tissue organization, and how to build simulations with particular mechanical properties. There is no textbook for this class. I was at a meeting in the fall, in December in the Netherlands where this topic has been discussed. Hayden has been working on a textbook, but at the moment there is no general textbook on the subject. And so that means it requires a little bit of discipline on your part because there isn't a single reference. There is a textbook that was written some years ago, about 10 years ago, by Shannon Preziosi that actually used our software in its incarnation of those days. The cover, actually, those pictures are from Computational. And it's not too bad, but it's a bit out of date. And so I'm not recommending it strongly as a necessary thing to do. What we do have are good online manuals, and I strongly recommend people dig through those manuals. And I also strongly recommend that if people find deficiencies in those manuals, they tell us. There are a lot of things that could be done better. This is a relatively small open source project, and we depend on people to tell us when things work and don't work. So we'd very much appreciate your help on that. We also have an archive on YouTube of this class from last year, the year before that, and so on, and all the slide decks from previous years. And in general, my plan is that the slide decks and lectures this semester will be pretty similar to the ones from 2023. And so I do strongly encourage people, if you're patient, to watch the video from last year's corresponding class. May not be exactly the same, but it will be similar. Or at least review the slide deck so that the ideas are familiar to you before we come into class. And that way we could spend a little bit more time doing exercises and working on ideas, a little bit less time on lecture. Okay. I sent around by email some basic material. I encourage people to print those out. There's a quick reference guide to Computational 3D. which lists a lot of the internal functions and basic operations. That'll make more sense by the end of the class today and starting tomorrow. For people who haven't used Python, there's a very basic Python cheat sheet that I sent out. There was a cheat sheet for a language called antimony and tellurium, which people who took my other class know about, which you won't need right away. And then online, there's also a reference manual. A lot of the things that we tend to do with this kind of modeling are related to developmental biology or to cancer biology. I encourage people to pick up a copy of the Gilbert Developmental Biology, preferably an old edition. The current editions cost hundreds of dollars and have a lot of molecular detail. One that's 15 years old, you can buy for $5 and has all the same biology in it, just less molecular names. Since we don't care so much about molecular names, you're better off with the old ones. It's got the biology is clearer. If you haven't had any mathematical biology, I like Nicholas Britton, Essential Mathematical Biology is a good basic math bio textbook. As I said, we've got some quick reference guides, the Computational quick reference guide, the Python cheat sheet, and Tellurium. We're not going to be using a very heavy-duty Python, but if you haven't used Python before, there's a little bit that's going to be getting used to. We certainly try to minimize the amount of programming that you have to do in this class. If people are interested in learning more about the methodologies that exist in Computational, there is a book chapter, which I wrote some years ago called Magnetization to Morphogenesis, which covers how this internal mathematics and physics of these models work. And there's also a copy of a paper that was written by a group of Canadian grad students called 10 Simple Rules for Tackling Your First Mathematical Models, a guide for graduate students by graduate students. It's not entirely what we need for this class, but it's quite good. Maybe somebody will be inspired and write a version of it for this class. There's also a draft of a version of it for this class that I sent around to people that you might look at too, about how to think about model development. And so I recommend people looking at that material as well. If you have not had Python before, I happen to like the online problems in Rosalind, which is a free online community for learning Python, taught around bioinformatics. I think it has really interesting exercises. There's some good introductory Python books. There are plenty of good online Python tutorials. There's no shortage. You really don't need a lot of Python. We also, in that archive of videos, we have a one-day Python bootcamp course that we teach every summer that covers what you need for this class in about 6 hours of lectures. And that also has exercises, and that's one way to learn it. How many people in the room have not used Python? Everybody's used some Python. Has anybody not used it at all? So you should be okay. If you know what a Python dictionary is, that's fundamentally the limit of what you need. The network modeling we teach a whole course on in the fall. We won't use a lot of it at this time, but there are demos for the parts we will use online. Again, we won't use a lot of mathematics. I try to minimize the amount of math we need in the class. If you're interested in biophysics, I really love the Phillips Physical Biology of the Cell textbook. It's a gorgeous textbook, but that's a reasonably heavy textbook, and so if you don't have a fairly strong physics background, it's not easy. An easier one is Physical Models of Living Systems by Phil Nelson, which is written to be a little bit less mathematical. And if you find anything that you think is really useful to learn any of these topics, physics, mathematics, computing, biology, networks, that you think we should distribute to the class, I'd appreciate you helping us with that. nobody comes into a class like this in general, if they haven't taken it before, knowing everything you need. Some people know more biology, some people need more computing, some people know more mathematics. But in general, this is a class where you need a lot of different disciplines at a not very high level of sophistication, rather than a class that digs into one specific thing in a lot of detail. It's maybe a little bit of an exaggeration to say you need a Wikipedia level understanding of a lot of different things, but it's sort of at that level. You don't need a lot, because the modeling that we're going to do is not terribly detailed. You don't need to know a lot of detail about the biology and the physics, but you do need to know enough to have a basic understanding of how biological systems work and how to translate them into computing. Okay. Any questions or comments so far on that? Makes sense. Okay, as a motivation, maybe this isn't the right audience for this, but these days we hear a lot about the need for data scientists and artificial intelligence, machine learning experts. Those are both great topics, and certainly we have no problem in our department and in the School of Informatics and in providing people with excellent education in those topics. But it's actually, if you're interested in going into an industry, or for that matter, academia, what we call mechanistic modeling, the kind of modeling that I teach, or Paul Macklin teaches, or Vikram Jadhav teaches, there's never really been a better time to do it. For example, the U.S. Food and Drug Administration now requires computer models of how drugs are distributed in the body, absorbed, and eliminated, what's called ADME modeling. And so every pharma company has to hire people to do that kind of modeling. There are newer companies like HeartFlow or Applied Biomath or Nova Discovery that use this kind of mechanistic modeling to do diagnostics, prediction, drug discovery, and so on. And so there really are a lot of opportunities for people to find positions in industry with this kind of training. The Embryo Institute up at Purdue I think most people in this class are already grad students, but there is an institute which has experimentalists and computing people devoted to this kind of systems biology approaches we're going to talk about. And they're always looking for grad students and postdocs. And so there's actually a big shortage of people with training in these areas. And so one problem that I find is that it's hard to get students to stay as PhD students or stay as postdocs because the opportunities in industry are quite good. And so I don't know whether people's ambitions are more academic or industrial, but in an engineering department, often people want to think about industrial careers. And this kind of training is pretty useful for that. We're in an engineering department because, and this is a slide that maybe doesn't fit as well in this class as in some other classes, because almost everything that we deal with in the modern world that's built, whether it's an airplane or a computer, is engineered. It's designed. It's designed on a computer. Biology is one of the few areas where we don't really design things. There aren't detailed models of how the body works to predict how your body will respond, for example, to drug treatments. And so if we're going to make health something that can be engineered, we have to be able to predict how health states change. And that's really a critical new technology. It often comes under the heading of digital twins. And so there's a lot of interest these days in, you hear a lot of talk about precision medicine, but in fact, without the ability to do computational prediction of health state and response to treatment, you can't really do precision medicine. And bioinformatics teaches you certain kinds of things, but the correlation between genotype and systemic response in some cases is there, certainly not absent, But in a lot of cases, there's so much feedback and so many levels of organization in a living organism that you really cannot predict from a molecular level interaction what will happen at the whole organism level from just looking at the molecules. And so bioinformatics is certainly necessary, but it's not sufficient to do this kind of prediction. And so there's a lot of interest then in developing this further. And so I think this is a topic, if you're interested as an academic, where one could spend one's life going forward easily on many of the questions that will be raised here. So we're going to be dealing with tissue organization. How do you build organisms? We're not going to answer these problems here in this semester, unfortunately, but we're going to start building the tools to answer those questions. How do organisms maintain themselves? This may not seem like a big question. In general, for example, the table that you're sitting at, I don't have to do anything to keep the table what it is, unless I break it. But in living organisms, these are dynamic systems. If you stop maintaining them, they fall apart. And that maintenance is actually rather problematic. It's not as well understood as you might expect. How do organisms fail? Well, one way they could fail is by injury. Another way they could fail is by infection. But one of the ways that people are particularly interested in is what are called developmental diseases. The classic developmental disease is cancer. If you look at cancer, it's often thought of as a genomic disease, but it's not really a genomic disease. It's a disease of misregulation of signaling. And in general, in cancer, what you have are normal developmental and homeostatic mechanisms, normal mechanisms that are used when organisms build themselves and maintain themselves that are being deployed in ways that cause problems. And so if you really want to understand how cancer works, you need to understand development and maintenance of tissues. And you need to understand a lot about the structural organization and interactions of cells. And there are certainly people in this department, for example, Feng Guo, who does organoid culture and organ on a chip experimental work. We'd like to be able to predictively design and build engineered living systems. And in principle, these kinds of mathematical models can be used to do that. And actually, two of my former grad students were just hired by a company in Washington, D.C. that's doing that. So it'll be interesting to see how that plays out. The kinds of questions that we might ask we've asked in the past using these kinds of methods would be, why do blood vessels sometimes invade the retina as we age, causing blindness? That's called choroidal neovascularization. And it's one of the leading causes of blindness in adults. Could we use the same kind of models to personalize treatments for diabetic retinopathy? Why do a subset of people have their kidneys grow uncontrollably until they die from kidney failure? Why do acetaminophen or Tylenol sometimes cause fatal liver disease at doses that seem to be non-threatening? Or how does a particular drug treatment affect the spread of a virus in a tissue? Those seem like very diverse questions, and they are, but they're all questions about how cells and their environment interact with each other and with the signaling inside of cells. And so all of these are questions that you need the same kind of basic infrastructure to address. And in this class, we're going to be teaching the basics, the basic language of how to begin to address these questions. Again, in one semester, we won't be able to probably do these higher level problems, but learning computing and learning to think about problems in a mathematical and computational way, it's like any language. There's a certain amount of investment that one has to make in learning grammar and vocabulary and conversation. You can't really write novels until you can write shorter works. And so here we're going to be learning the basics. in the hope that if you're interested, you can then go on and do these more advanced topics. So what are virtual tissues? If I look experimentally in biology, these are all developmental biology simulant movies. The one here on the bottom left was taken actually by Maria Bonderson, who's on the faculty here. What we see is that there's a lot of movement. And in the top left, we see branching morphogenesis, the formation of the branching structure that leads to the lungs, the branching structure in the lungs and in the kidneys. In the top right, we see a movie where they've actually been able to make a beautiful 3D movie of all of the cells in a developing fish embryo during early development. The bottom left, what you're seeing are actually the blood vessels also in a fish embryo that are growing between what are going to be the ribs of the fish, what are called intersegmental vessels. And you can see them actually growing out, reaching out and growing. And then on the bottom right is the also early development. This is a movie of a chicken embryo. Chicken embryos are nice because they're flat and they can be grown just in a dish without putting them in special culture conditions. And what you see are the formation of these little balls here, which are called somites, and those are going to give rise to not the vertebrae themselves, but are going to reorganize and eventually give rise to the vertebrae in the animal. These are all problems that have been worked on computationally using either a Computational or similar modeling methodologies. We're going to be focusing on the world as seen from the cell level. We're going to think of cells as active agents. In biology, at the tissue level, cells really are the things that are doing things. They behave as individual agents. We're going to think about how those cells are controlled. So we're going to think about, at least later in the class, we'll think about networks that are controlling the behavior of the cells. We're going to think about how those cells interact with each other and with their environment, in particular their biochemical environment. And then sometimes we'll put all of those tissues inside a model of blood flow and transport in the whole body, what are called, again, PBPK, pharmacokinetic models. But our models will always have the cells in them, and they'll always have the extracellular environment in them. Sometimes they'll have the control level, sometimes they'll have that higher level as well. So these are all intrinsically multicellular and multi-scale models. This is a little simulation of that chick process of segmentation that we were seeing experimentally in that movie. The colors in the left are oscillations of a particular gene in the embryonic cells. The gene has a wonderful name of lunatic fringe. In the middle is an extracellular gradient of a signaling molecule called fibroblast growth factor, which controls cell differentiation. And then on the left, you're seeing the cells differentiate and reorganize in response to those signals, the combination of intracellular signals and extracellular signals. And so this is a simulation, actually, of segmentation. These are mechanistic agent-based models. What we're going to be producing typically are movies. The simulations are going to give you movies as outputs, sometimes in 2 dimensions, sometimes in 3 dimensions. They're based on hypotheses about mechanism. How do the things work in the organism? What are the fundamental interactions, behaviors, processes operating in the organism? They're additive. So it's the opposite of data science. In data science, you start with large data sets and then you apply filters to pull out information. In this case, we start with nothing and build. So it's the opposite direction from what typically you do in data science. We're focusing on cells to organism, not on what's happening inside individual cells. And they're based on quantitative models of what objects do, what cells do. When are cells created or born? When do they divide? When do they die? What forces do they apply to each other? How do they move? So these are the kinds of questions we're going to be asking. It's not easy to build these models. There are a lot of things that stand between us and predictive models of biology. We're talking about very complex systems. The science is not very well understood. There are lots of different types of cells and lots of different types of components. They do a lot of different things. In industry, if we have two screws of a particular size, we want them to be as mechanically identical as possible. Two cells of the same type are different. They're not ever the same. And so that makes the abstraction from biological reality to model a little bit more challenging. We certainly can't describe biological processes in complete detail, and it's not going to come up so much in this course, but when you start building applications, you'll find that comparing experiments and simulations is a little bit problematic. For one thing, biology is stochastic. There's a lot of randomness in biological processes. And so that means that even if I start with a simulation and an experiment with identical initial conditions, and even if the simulation were perfect, I would not expect that every cell in the simulation would track every cell in the experiment. That's not realistic to do. It's not like quantum mechanical uncertainty, but nevertheless, that kind of uncertainty is there. And so there are a lot of issues that we have to address. Surprisingly, though, you could do pretty well. You could actually explain a lot of biology and a lot of medicine. And you can make predictions that are used in industry and in clinical practice using this kind of modeling. There are a lot of different modeling methodologies that are used to represent cells. They differ in their level of spatial detail, in the speed at which they run. None of them are perfect. For example, Paul Macklin here at IU has a great package called PhysiCell, which is based on essentially molecular dynamics models where cells are point-like objects. They're spheres. In Computational, which we'll use, cells have explicit shapes. That gives you more detail, but also means you can't simulate as many cells. And so there is a whole zoology of different kinds of modeling approaches. We have some PhysiCell simulations up at the top. Biocellular, these are all point particle methods. Simulation packages like EpiSim, Biocellian, CellSys, Chaste, which are pretty well developed. Former member of my group, TJ Seago, has a package that does this called Tissue Forge. It's very nice as well. And then there are methodologies that have more explicit shapes. There's some essentially finite element mechanics models like Cardiodocus's approaches, which give you a lot of biomechanical detail, but are very computationally expensive. There are what are called vertex models, which are used particularly in certain kinds of developmental processes. And so there are a whole variety of different approaches to doing the modeling computationally. We're not going to focus here on what's happening under the hood, but sometimes we'll have to be aware of that because it'll influence what we do.
Can I ask a question?
Please, go ahead, Kyle.
Do you run up against the wall of the computational intensity or the memory or size of the models when you're doing these things? Where are the roadblocks or things that get in the way?
So for the people who are online, Kyle asks, when we do this kind of simulation, what limits the size of the simulation? Is it memory? Is it compute time? Is it something else? And this varies a little bit depending on the modeling methodology you're using. The methodology we use in Computational is pretty fast, but it's memory hungry. Simulation packages like PhysiCell or Tissue Forge, which are center-based, are much more memory efficient, at least for the cells, although not so much for the representation of chemical fields. In that case, you almost always have to discretize it on a lattice. And so they tend to be more compute bound. Some of the other methodologies tend to be, the biomechanical ones, the finite element ones tend to be pretty compute heavy and quite expensive computationally. So there always are trade-offs. simulation package like TissueForge can simulate about 1 million cells in a reasonable amount of time. Physicell can do, I think, about 100,000 cells. Computacell, you're talking about thousands of cells to 10,000 cells. Essentially, you get about the same level of you could have about as much spatial detail in any of these models before things get overwhelming. And so in this case, we've decided to represent the spatial structure of the cells, and that means we can't model as many cells. Good question. Computacell itself consists of an editing suite called Twedit++, which is an editor that has some smart features to try to help you learn how to build computational models more easily. And then an execution environment called Player, where you run the simulations and visualize them. In some years, in fact, in last year, we used an online version of Computacell in NanoHub. The online version is a little bit old, and so this year I hope that people could use the desktop version instead. There are a lot of basic biological and physical concepts that are supported by these kinds of modeling frameworks. We'll hear a lot of talk about objects, processes, dynamics, and initial and boundary conditions. Objects are the things that are being modeled. In general, we're going to be talking about cells or generalized cells. Those are volumes of material that do particular things. We'll have a concept of fields, continuously varying processes. Those could be chemical concentrations, they could be gravitational fields or electrical fields. There's a concept of links. Those are rigid objects in the world, springs, essentially. And in a non-spatial context, networks of control. Each one of these object classes will have its own set of properties, its own set of behaviors and interactions. And we're going to have to learn what those are and how to control them. So let's actually try doing a quick exercise. So if people could get their computers out, why don't we see if we could do a quick use of Computacell? And how many people were able to download and install Computacell on their computers? Okay, everybody. Okay, how about our online friends? Everybody got it running. Even the Mac people got it running. That's amazing. Usually the Mac people is an issue, their issues. So I'm glad things are working for them. Windows is pretty easy for the installs. Mac somehow are a little bit harder. Okay, does anybody, so people don't need, we have in the room, we have Pedro who's taking the class, but actually is pretty experienced. And so if we need rapid debugging, Pedro's your man. Okay. So let's, if everybody says Computacell up and running, let's do something very quick, which is we're going to just run a simulation that Computacell comes packaged with a set of what are called demonstrations, which could probably be organized a little bit better than they are. Promise they could be organized better than they are. But we're going to be talking about how blood vessels form. Blood vessel formation can be called angiogenesis or vasculogenesis. Those two terms represent slightly different ways of organizing blood vessels. The blood vessels themselves are composed of what are called endothelial cells, primarily endothelial cells, it's a particular cell type in the body. But the big blood vessels in your body, like the aorta, and peripheral blood vessels like capillaries, actually formed by different mechanisms. And so it was quite interesting that the same cell type is building the same structure, but building it in different ways in different contexts. And so this was a topic that was of great interest 15 years ago or so, 17 years ago now. And so I was quite lucky. Actually, 20 years ago, Roland, because the first one was in 2005. So I was very lucky that 20 years ago, I had as a postdoc in my group Roland Merks, who's now professor of biomathematics at the University of Leiden in the Netherlands, uh, who was interested in modeling blood vessels. And so we did a series of papers on this topic. And the basic question is, starting with these endothelial cells, how do the endothelial cells signal and move and organize to form capillary networks? And that's called vasculogenesis, uh, is what happens when you have uh, distributed blood endothelial cells that spontaneously organize into networks. And that's what happens in the peripheral vasculature. And angiogenesis is what happens when you have sprouting from existing blood vessels, for example, with the aorta. Um, After you have that initial formation of the strut network of the capillaries, the cells themselves change shape. They hollow out to make essentially tubes, and then there's remodeling and what's called pruning to form full vasculature. But this is looking at the very early stages of vasculature. And we wanted to understand if the mechanisms were the same. And there were some experiments that had been done which suggested that the cells secrete a chemical called vascular endothelial growth factor A, VEGF-A, which is pretty well known if you study cancer, because cancer tumors induce the growth of blood vessels to supply the tumor with oxygen and nutrients. And so VEGF-A is a chemical that's used in normal maintenance of tissues, but it is also deployed in a pathological way in cancer. And they observed that if you have gradients of VEGF-A, that the endothelial cells crawl up the gradients. They move up gradients of VEGF-A. That's called chemotaxis. There were also experiments done by our collaborators in Kansas University Medical Center that suggested that there were other things that were going on, in particular that when the cells touch each other, they have molecules on their surface that mean that the surfaces that are in contact with other cells don't respond to the extracellular chemical. And so that was the basic experimental hypotheses that we were working from. And so Roland built a simple simulation where we have cells. The cells secrete a chemical, VEGF-A. That chemical diffuses in the environment. It decays in the environment. The cells measure the gradient of that chemical and move towards higher concentrations of the chemical. And if I look at these two simulations, and I'll play this movies again, the one on the left and the one on the right start with the same initial conditions. But in the left, all surfaces of the cell respond to the chemical. While the one on the right, only the free surfaces of the cell respond to the chemical. And you get this totally different patterning. In the one case, you get blobs, which is observed experimentally if you knock out this particular factor that prevents chemical response on contact. On the right, the cells organize to form struts. On the top right, you actually see that process in action, and I'll play it one more time. That process in action in the lab, this is by Ambrosi et al., where they took endothelial cells, put them in culture, and they spontaneously organized to form vascular patterns. And when I had a wet lab before COVID, actually, that was an experiment we did in my lab quite a bit, that particular vascular patterning experiment. In fact, we have some patents on that, on some ways of doing that. And so the key idea is here that we have cells. The cells secrete a chemical, and they respond to that chemical, and they respond differently on their free surfaces and on surfaces with contact. Okay, and now we're going to run a simulation of that. We're not going to worry right at the moment about how the simulation is built, but we're going to run it. And so I hope everybody says they have Computational running. If I do a split screen, so that I have Computational running on one side and the slides on the other side. That's nice in some ways, but it also means that if people are looking at the screen on a laptop, it can get pretty small. So maybe for this one, I should just I'll walk through the instructions and then I can do it once with the slides full size, and then we can go back and do it again. So is there anybody who does not have Computational running at the moment? Everybody has. Okay, so I can skip the installation instructions. And what we're going to focus on is using Computational Player at the moment. So as I mentioned, Computational consists of two parts. One is a model editor called Twedit, and one is Computational Player itself. And since we're only running a simulation, not editing it, we'll just use Player. And so the first thing to do is to launch Computational Player. And if you're on a PC, you would go to the search bar and type in Computational and launch it. If you are on a Mac, you have to find the Computational icon and launch it. And so I'd like everybody then to launch Computational and give me a thumbs up online or in the classroom when you've got Computational running. And what you should get is something that looks like this. It'll be a window. It won't have anything in it, which is Computational Player. Alex says it works. Well, I know you know how to do this, so it's not it's not, but it's but I want to make sure I'm not going to go on till everybody has it working. So, Santu, Jonathan, and the people in the room. William, do you have it?
I mean, I'm doing the online version.
Oh, you're doing the online one. Okay. So I'm going to go forward just for a second. I'll come back and do this again. Once Computational is launched, it's a window. You can resize it the usual ways, pulling on the corners and so on. One thing that you may find that you want to do is that when it opens the first time, you'll see a box called Model Editor and a gray window here. The gray window is where everything interesting happens, so you may want to slide this bar over to give yourself more space. Okay. And then to open a simulation in Player, you go up to the File pull-down menu. You select Open Simulation File. And if Computational is configured right, it should give you point by default to a directory which has Computational demos in it. And I'd like people to try doing that. If you don't point to Computational demos, you're going to have to find where you installed Computational. And you should find Computational demos. And the indexing is not the greatest. So the title of the demo is Computational 3D Demos, Book Chapter Demos, Computational Methods in Cell Biology. It's a mouthful. And then /angiogenesis. And if you open that, you should find a file called angiogenesis.cc3d. Double click on that and hit open. And then hit play in Player, and the simulation should run. And I will once we get everybody up to speed, I will come back and do this with you. But if you've got Computational running, you could try that out. If you have any trouble finding that folder, or if once you're in the folder it doesn't run for you, let me know, and I'll do my best to help out. One thing, if you're so there are one thing that I gained by doing things locally rather than on NanoHub is that there are a certain number of annoyances on NanoHub. One of them is that you will actually have on NanoHub 2 copies of the demo folder. One copy will be in the root Computational directory, and one copy will be in your local scratch space. And you have to make sure that you navigate to your local scratch space and open it there. If you open it in the root directory, the Computational will crash because you don't have write privilege. Yes, no, Pedro?
It's installing now.
Okay,
It may take a while.
okay. Okay, what about other people? Were people able, did it run, not run? William, how about you? Pedro, could you maybe look over William's shoulder? Jonathan, it ran for you?
Nice, but beyond.
Alex, worked for you. Once we've got it to work once, it's pretty easy. So we'll take a little bit of time to get everybody working, and then once it works one time, it should be easier. If you've got it running, there are a couple of things, and again, I'll come back for people who are waiting. There are four control buttons in Player that look like old-fashioned cassette or CD player buttons. A run button, a play button, which runs the simulation. The thing that would have been a record button in the old days, the circle, which does a single step. The pause button, which pauses execution but allows you to restart, and stop, which terminates simulation execution. And if you hit run again, it starts from the beginning. And so I strongly recommend that you don't hit the stop button unless you know you've made a mistake. Use the pause button when you need it, just to halt the simulation. And we'll come back to it. How's the install going there? Still grinding away? Yeah, the first time we changed the installer for Computational, and the new installer should be more reliable, but it's slow. So what happened, Pedro?
We cannot access the demos folder. Nothing shows in the browser, in the file
Huh.
browser.
So
Yes. E4 manageable from that.
Yeah, this was the one we
No,
were.
no, it's not.
No?
No, I used the different one.
Okay, so let's try.
So there is also the angiogenesis tool in which you can run by itself. That may be the one to use.
That was what I was going to do, but
Yeah. Change them up.
Again, I appreciate people's patience while we get everybody started. If you've got it running, you can play around with visualization. There are a couple of things that are useful to know. You should have a simulation that looks like this to begin with. That's a section through the simulation. It's a 3D simulation. If you pull down here in the graphics window where it says XY, you could say 3D. You should be able to visualize in 3D, and then you can rotate and pan and zoom. If you have multiple windows on PCs and on Linux, but unfortunately not on Macs, there's a function called tile that will lay out the graphics windows for you. Unfortunately, the Mac graphics capabilities doesn't allow you to do that. And so I would say, why don't you people just play around with getting running the simulation a little bit and with visualization. If you make a mess, if you hover over the screen and hit the letter R for reset, it will resize everything and put it back the way you started. It should be called angiogenesis. So what's happening is the demos are not loaded in the.
No, well, no, I initially used the wrong tool on Navajo, but now this
Yeah.
one I think is works, right?
That's
So
yeah, there you
here,
go there. Yeah.
here,
Demos, yeah.
and then, I don't know.
Book chapter demos.
Oh,
Angiogenesis, but you're in home. You have to double click on
Oh.
yourself. There you go. Now do the same thing.
so just go.
Yeah,
all
there,
the way.
demos, same thing. All right.
Oops.
No, up. Book chapter demos, angiogenesis, there you go.
Thanks.
And you can do everything from that angiogenesis demo. It has all the capabilities as the one that's as the V4. So it should give, if you go to the install page, it gives you instructions, but I have to say that the instructions are correct, but they're not necessarily easy to follow. That mini Conda installer is not the easiest thing in the world to use. And the reason we went to the mini Conda installer was because we were having too many clashes with incompatible libraries. And with the Conda installer in principle, then you're supposed to be better in terms of consistency. But the problem then is it downloads everything, which is why the install takes so long. I don't think there's any ideal solution for Python packages. It's just it's an issue. Okay, so that's something I should have mentioned. If you really get stuck, the way William was stuck, if you go to under visualization, under the visualization window, there's a reset camera for graphics window option. Yeah, but it's the command does. Let me see if I could show you a little bit about what to do in terms of controlling the simulation. So one of the things that you could do is you could change the visualization. So under visualization, you can turn cells, cell borders, and so on and off. In 3D, cell borders often are something you don't need. If you want to control what it looks like, under Tools, or here, this little this little screwdriver and wrench symbol will pull up a CC3D preferences window. You have the option of controlling the colors of your simulation here. And in 3D, you can control what cells are visualized and what aren't. While we're waiting for other people to get things installed, you can try playing with the colors. Something that's more interesting is you can create additional graphics windows. This button here that shows two graphics windows, one behind the other, or under window, you can pull new graphics window down. It'll create a second graphics window. And then within the window, you could select the chemical field, VEGF, here, instead of the cells. And you can visualize the chemical that's being secreted by the cells. And so why don't people play with that a little bit and explore that while we're making sure that everybody has a working version of Computational. So I'm going to do that myself. So here's my Computational window here. I resize a little bit. I'm going to go to File, Open Simulation, Demos, Book Chapters, Angiogenesis. Now I'm going to run it. I'm going to go to Window, Tile to lay out the screen for my. And here I have the cells on the right and and also cells on the left. I'm going to change that one to be. I got a point on it. I'm going to change that one to be the chemical field. What is it doing to me? This is another. This isn't okay. This is an issue with. I'm going to change it to be VEGF. And so now I can visualize the chemical concentration and the cells together. I can make this 3D as well. And so I can see the chemical concentrations and the fields at the same time.
What will the isosurfaces look like?
Well, you'll see basically tubes around the cells, which will represent the positions of the chemical concentration. So it'll draw isosurfaces. It's actually pretty nice. Alex, can you screen share?
The only weird thing I have is it shows up on my other screen. So when I click this, I see it. It's just off on the other screen. And then what it'll change is then once I click it, it goes back. It'll change. I don't know. There's something weird with this. I need to try looking at it later with only one.
Well, occasionally, Computational and Zoom don't play nice with each other. But that looks, I mean, that looks basically like it's working for you. You're able to, it's running, you're able to visualize the chemical concentration, you're able to do 2D and 3D, you're able to change the viewing. So that looks all right. Jonathan, did it work for you?
I had everything working, and then I clicked ISO while you were talking about it, curious, and now everything's crashing out on me.
Okay, so what to do? So relaunch it.
Yeah. Here, I can
Let's see
just
if we can
share
unclick
my-
the isosurface thing. And
That's the problem. I cannot.
Okay, why don't you screen share? Let's look at it together, see if I can get that. See if
Are you seeing?
Yeah.
So
Yeah.
I'm in field, but it's all grayed out. Not allowing
Okay,
me access.
so why don't you hit step, the step button, the circle, the like the record button, that one. Hit that. Hit step. No, it crashed. Okay, let's try running it one more time. And now let's open a different simulation. Try to get back. So, oh yeah, restore default might work for you.
I already tried that. I could try it
Okay,
again, but
so there's
default
a
global or for current project.
Global settings. And let's see
Oh,
if
it's not opening recent anymore.
yeah, let's hope that there's an annoying hidden file which has the configuration, and we'll have to get rid of that. Did it crash on you or is it opening? Yeah,
Yeah, destroyed.
Okay, let's try one more thing. Let's launch player again.
Okay.
And now go to file and let's open a different simulation.
Do this.
Go to demos, do a book chapter, do cell sorting. CC, okay, now let's run that, see if that'll launch.
So try installing only player 5.
See if we hit play, does that work? Okay, so that works, okay. So now let's go to visualization.
No, this is the only one.
Sorry, it's not visualization, go to config, yeah.
Which is
Okay, it's not, so okay, so now let's try going back to the other simulation, see if it'll work.
but it has more steps.
Okay.
Do I need to exit out of this one? I
Oh,
guess.
it should just hit stop. Hit the stop button. Okay, now open back to the angio. Yeah. And just hit step now.
Oh, sorry. I pushed start and it seems like
That's all right, but it's okay
it
now.
went.
That cleared.
Okay.
So sometimes things are it tries to be too smart. So what it happens is the Computational will save the configuration of player,
Mm-hmm.
so that when you launch it again, it continues with what you did, so you don't have to set everything up again. But the problem is if you wind up with an illegal configuration in player, then that could cause that crash. And there are instructions online, and also we'll have in class for how to there's a hidden file, which has basically it's a JSON file that contains sorry, it's an SQLite file. I can say JSON file. It's an SQLite file that contains the player configurations. And the problem is it's a hidden file, so you have to find it to delete it. If you delete that, then it'll restore. But you're okay now. Anybody else, Santu? How about you? Is it working for you? Great. So you've got that working too.
Yeah.
Okay, great. Okay, you check because there's a new release candidate, which we're not using because I didn't want to use a beta version. I hope it looked like everybody got the basic simulation to run, and we're able to do a little bit of control of color and things. And I apologize to people who had issues with getting it up. But it took us what, an hour? We're an hour and 45 minutes into the class, so it's not ideal, but I guess it could be worse. uh, uh, so I was going to spend a little bit of time going over so quickly some of the applications that Computacell's been used for over the years. And then I was going to show you a little bit about how you code the simulations. So Computacell's been used a lot for basically basic developmental biology and studies of basic biological mechanisms. It's been used a fair amount in toxicity studies, in toxicology, computational toxicology. It's been used to some extent to design new therapies and to personalize therapies. And you can always ask the question with any computational methodology about, when do you need it, when don't you need it? When you're modeling the heart, there are very, very sophisticated modeling packages for modeling electrophysiology of the heart. I'd say much more sophisticated than anything in Computational. But the heart has complicated mechanical and electrical properties, but the cells in the heart don't move relative to each other. They're fixed relative to each other. So the heart behaves like a big, complex, solid object. When you're doing drug distribution in the body using what are called pharmacokinetic models, you have a lot of complex transport and a lot of complex metabolism, but you don't have individual cells usually. And so those are examples where you have different things that you're trying to analyze, and so you use different modeling methodologies for them. If you're going to be modeling radiation dosimetry for cancer, you have to model volumes of tissue that are centimeters or tens of centimeters across. You're not going to be able to model every cell in a 10 centimeter cubed volume because they're 10 to the 9th cells. You can't do it. And so, again, we're going to find that depending on the kinds of problems you're trying to address, you're going to use different kinds of methods. On the other hand, Computacell or a program like Physicell, Paul's Physicell, or these others, do allow you to build these wide variety of biological simulations. This is a very beautiful simulation, which I wish we had as a demo in Computacell, that was developed by Giulio Belmonte talking about hair cell organization in the Drosophila wing. In this case, probably should have been at the end because it's a rather sophisticated Computacell simulation where you're actually modeling the reorganization of chemicals within individual cells and how that leads to polarization of the cells. This is a simulation that involves using subcellular modeling, Delta-Notch, which is a signaling mechanism that we'll run into a couple of times during the course. When you're simulating networks, you can simulate networks in a variety of ways. We tend to use reaction kinetic models, but there are also what are called stochastic Boolean models of gene regulation, and Computational supports those as well. In this case, the signals between the cells are controlling the cell cycle and the growth and division of the cells. In this case, we're modeling only one cell, but we're modeling in much more detail what's happening inside of single cell. In this case, the cell's in 3D. It has cytoplasm, it has a nuclear compartment, and it has a leading edge, which is simulating actin polymerization and cell crawling. And so this is a rather detailed model of cell crawling on a substrate of a single cell that was developed actually by Pedro's old advisor back in Brazil, Gilberto. And so that's an example of putting a lot of spatial detail into a single cell in a simulation. This is a classic simulation which we did many, many years ago, which I rather regret not following up on, which was trying to understand blood vessel development and its interaction with tumors. In this case, we have a simulation of a tumor spheroid. The cells in the tumor consume nutrient and oxygen. The cells grow. As the tumor gets bigger, the cells in the center of the tumor are oxygen deprived. When cells don't have enough oxygen, they send out that VEGF signal that we were just looking at a few minutes ago to induce proliferation of blood vessels. And so what happens then is that the blood vessel network begins to grow and supply additional nutrient and oxygen to the tumor. In the top simulation, we have normal proliferation of the blood vessels and growth of the tumor. In the bottom simulation, we're simulating what happens when you use an anti-angiogenic drug like Avastin, which prevents blood vessel proliferation. The tumor still grows, but it grows more slowly. And interestingly, it moves fast. It actually migrates through the body more rapidly. And there is some evidence of more metastasis when you use anti-angiogenics as cancer therapies. And so this was a rather detailed study of the phases of tumor growth in the presence or absence of neoangiogenesis. This was a simulation of what's called somatic evolution in tumors. Tumors go through what are called cancer progression. Once you have inappropriate proliferation of cells, you get a benign tumor. It doesn't become a cancer until a variety of things happen. The cells have to escape immune surveillance. They have to be able to migrate out of the initial tumor and form additional metastases. And you can ask the question, people think of cancers again as genetic diseases where there are particular mutations, but you can have a mutation that would reduce the rate of cell proliferation instead of increase it. You could have a mutation that would make cells less likely to metastasize rather than more likely to metastasize. And so you can ask the question, if it's possible to increase or decrease a certain behavior, what is it about the environment that selects you for the ones that are adverse? This is an approach to understanding cancer, often called cancer's ecosystem or somatic evolution, that was pioneered by Bob Gatenby at Moffitt Cancer Institute in Florida. And it's led to really radical changes in the way people think about cancer and cancer treatment. This is a very simple simulation, also by Gilberto, in which we simulated cells that could change their adhesion relative to their environment. Randomly, when the cells divide, it's possible for them to become more adhesive or less adhesive either to each other or to the environment. We were very careful to make that mutation neutral so that if there were no selection, the average adhesion wouldn't change. And so then any change in adhesivity that you see is due to selection pressure in the environment. And you could show that under certain conditions, there was positive selection for the cells adhering less to each other and more to the environment, which made them more likely to metastasize. And so in this case, you could show that there was, in fact, environmental pressure due to the structure of the system itself that led to metastatic behavior. So that's the kind of, it's rather abstract. It's certainly not a full cancer model, but you could show that some of the things that you saw in cancer progression could be understood. I mentioned choroidal neovascularization. This is another vascular simulation. This was also done by Abbas Sharinafard, who's now a permanent staff at St. Jude Children's Hospital in Memphis. Does a lot of modeling and experimental work there. One of my first graduate students when I came here to IU. And he did some really beautiful simulations of angiogenesis. In this case, we have a model of the retina, the backside of the retina. We have the blood vessels behind the retina, called the choriocapillaris, which are the densest blood vessel network in the body. We have a model of the pigmented retinal cells, the black cells that line the eye, and the photoreceptors. And in some individuals, as you age, there is an invasion of the retina by blood vessels from behind the retina, and that could lead to retinal detachment, breakup of the retina, and blindness, which is what you're seeing here. You're seeing invasion of the retina, detachment of the retina, then eventually breakup and death of the cells of the retina. And in this case, we were trying to understand the mechanisms that led to that and what it meant biomedically. And that was actually rather a nice simulation because when we did it, the theory about what was happening in that disease was a growth factor theory. We showed that from computational point of view, that didn't seem to be likely to be the case. And later on, it was actually experimentally validated that, in fact, the main problem was adhesion defects in the retina. Segmentation we've seen, that was our sort of base case. These are simulations that were done by Giulio Belmonte, who's now on the faculty at North Carolina State University. We did these simulations based on what was understood about the signaling networks inside of the cells that are going to give rise to the somites. That was that movie of the chick that we saw before. Here, the organization of the somites here, and we saw the simulation of that movie. And in this case, we have these complicated intracellular chemical oscillators that are interacting with this long-range chemical gradient that lead to differentiation of the cells and organization. Our experimental collaborator, Claudio Stern at University College London, however, showed that he could knock out this entire network of oscillations and get rid of the gradient, and you still got somites. So we had this beautiful computational model that explained what was understood, but then the experimental facts changed. In this case, Giulio was able to show that there was an alternative, purely mechanical mechanism that could give rise to somite organization, where the cells polarize, they co-align the polarization, and that leads to spontaneous mechanical organization of somites. And that was really quite a pretty simulation. These are simulations where the cells have compartments and rather complicated mechanical properties. So this is pushing CompuCell in the direction of more detailed mechanical simulations. We worked extensively over the years with Bob Bacalhau, who is a surgeon at the medical school, on polycystic kidney disease. This is a relatively, fortunately, rare disease where there's a mutation in a ciliary protein, PKD1 or PKD2 associated with it, that leads to your kidneys developing cysts and growing until you die, when your kidneys are the size of watermelons. It's currently untreatable except by kidney transplant. There are no drugs, therapies for it. What was not understood was how the mutation led to the cystic phenotype. The mutation is well understood. If you have that mutation, you're almost certainly going to get the disease. If you don't have that mutation, you're probably getting the disease is less than 1 in a million. But the mutation in the cilia didn't seem to correspond to the mechanical properties of the disease. Bob Bacalhau identified that mutation in the ciliary protein caused in a few cells of the kidney the expression of an incorrect cell adhesion molecule. And we were then able to simulate the formation of a kidney tubule, the signaling between cells of the kidney tubule, introduce a single cell with the incorrect signaling molecule, and show that that led to a cystic phenotype, which under certain hypotheses then matched what was seen experimentally. And so this was actually simulating in vitro experiments. At the bottom, you see actually experiments in human kidney 1 cell line that Bob did, where he took normal human kidney cells. They produce normal tubes. You transfect a small number of those cells with the wrong cadherin, the wrong cell adhesion molecule, and that induces the formation of cysts, which corresponds exactly to what we saw in the simulation. And that went on then to do some drug discovery, and Bob is currently trying to commercialize some drug targets for that disease. These are simulations also by Giulio Belmonte, more abstract simulations about 3D biomechanics. In early embryos, you get elongation of tissues due to contraction, directional contraction in the tissue. And these were some simulations that were done by that. That's a simulation we could definitely do in the class this year. During the COVID period, people said we'd rather do viral infection rather than cancer models. So we put together during the class that happened in March in this class. It was March 2020. The students said, "Hey, we'd like to do viral infection models rather than COVID, rather than cancer." A group of the students did a class project on building a simple viral infection immune response model. We kept working on that with some postdocs, and actually we published it in December of 2020. So that was pretty fast. So that was actually sort of a nice experience for us. So in this case, we have a little patch of epithelial cell modeling the lung. We start out with a single infected cell in the middle of that epithelium. That infected cell produces additional virus, which spreads and infects other cells. It pulls in immune cells. The immune cells try to control the infection. In the particular image that I'm showing here, the immune cells aren't responding fast enough, and the infection will kill all the cells in the patch of tissue. Giuliano then went on and tried to model in more detail what happens in people have forgotten remdesivir, but it was the first antiviral for COVID. Trying to model how remdesivir works in the body and controls or fails to control the infection, and did a set of rather detailed studies modeling the dosimetry of remdesivir and the effectiveness of control. That initial simulation had a very oversimplified model of the immune system. T.J. Sigo, who's now on the faculty at University of Florida, took that initial model, and there was a relatively well-validated, quite detailed non-spatial model of immune response to influenza in mice by Bard Ermentrout and Erica Mohan. He put that into a spatial context and built this very beautiful model of influenza infection and immune response, which now has macrophages, natural killer cells, CD8 plus T cells, CD4 plus T cells, B cells, antigen-presenting cells, and eight different cytokines. And so this is it took some time. It took 2 years from the initial simulation that we did with the students. But this was actually trying to build a serious model of immune response in tissue. We worked for a number of years on diabetic retinopathy with people here in the School of Optometry. In the choroidal neovascularization, you have blood vessels that are invading the retina, In diabetic retinopathy, typically you have the opposite problem. The blood vessels are dying when you need them. And for a long time, this was treated by going in and killing the photoreceptors to reduce the oxygen load of the retina. And that's a pretty ugly treatment strategy because you're destroying the photoreceptors in your eye to try to save the eye. It's not totally crazy. This is very much like fighting forest fires by building fire breaks. You're essentially trying to build fire breaks. And of course, when you cut down trees to build a fire break, you're also doing damage. Or sometimes even in cities, you'll demolish buildings in the path of a fire to try to prevent the fire from spreading. The problem was that the fire breaks in this case, which were trying to prevent the spread of the disease, were being put down essentially randomly. And that meant that sometimes they were ineffective at controlling the disease, and you were doing a lot more damage than you needed. And so we were modeling what we believed were the mechanisms of progression of the disease, and then trying to design optimal targets so that you could control the spread of the disease more effectively with doing less damage. I mentioned toxicity. We've worked a lot in toxicity over the years. Acetaminophen, of course, Tylenol, is one of the classic painkillers and antifibretics that's used. And there are certainly situations in which it's indicated, but it's given as an over-the-counter medication, and it has one of the lowest therapeutic indices of any medication that's used commonly. It's the ratio between the dose that you take to treat your headache and the dose that will kill you is, for most medications, if you swallow a bottle of aspirin, you'll get an upset stomach. You swallow a bottle of ibuprofen, you may have some mild kidney damage, and you'll certainly feel sick. You swallow a bottle of Tylenol, you will die. And one of the problems with Tylenol is that the lethal dose varies depending on things like, did you have grapefruit juice in the past day? Or did you consume alcohol? Or did you eat? And so every 15 or 20 years, it's rediscovered that a bunch of fraternity boys drink heavily. They wake up in the morning, they haven't eaten, they take 4 Tylenol, and they die of liver failure. And then it comes on then there are congressional hearings about it. It's in Consumer Reports. They talk about banning Tylenol, and then they decide it's too useful. They forget about it again. The EPA actually came to us to talk about whether they could ban Tylenol as a pollutant. But to do that, they had to understand the mechanisms of damage. And so we did a series of experiments here with people in the School of Public Health and then simulations where we were trying to understand how Tylenol is transported in the liver, how it does damage, what are the effectors that cause some people to be more vulnerable and when. That project of ours was trying to understand population variability. Our project ended, but there's a European project that's continuing today in the ONTOX program led by Dirk Dresdo, who has his own modeling methodologies, where he now has extremely detailed models of the liver and transport in the liver, trying to look at toxicity patterns in the liver. There are also people, Bhattacharya at Michigan State University has worked extensively and published using Computational on modeling liver toxicity. And so those are some applications there. So I thought those would give you a few ideas about different things that people have done. Maybe we can hear for a few minutes, since I try not to lecture for too long, maybe people could tell us a little bit about your background and interest, just 2 or 3 minutes, if it's okay with you. Maybe start with the people online. Santu, could you tell us a little bit about yourself?
Yes, sir. So I have did my undergraduation in biotechnology. I have no experience in computers, but I've been recently learning Python, and my major in grad school is bioinformatics. So I'm still trying to explore different streams and areas in this biology and informatics departments,
Mhm.
and I would like to learn more about the career aspects in this area.
Great, thank you. That's fine. Okay, go ahead.
Trying to put the Zoom screen on the.
Oh, I'm sorry. Yes, thank you. I forget what I see and what the people in the and I should ask people if you're going to talk online, I should probably ask you to turn your camera on so we can see. Oops, sorry. Um, is that okay there? Jonathan, do you want to tell us a little bit about yourself? He may have given up. All right.
There we go. Hello. Hi.
Okay.
Can you hear me? Yeah.
Oh, there you are. Okay. Okay.
I am a master's student in biotechnology. I did my undergrad in microbiology. And this semester I'm taking a whole bunch of electives focused on more bioinformatics and computing and kind of that crossover between technology and biology. Yeah, I don't have a ton of coding experience. I did some stuff with Java in high school, but Python's newer to me, so I'll probably be doing those tutorials. So
I mean, again, one of the things that we have in this class is a lot of variation in background and interest. And when, provided there are enough people who stay in the class, we try to, when people do projects, we try to team people up. So somebody with a more biological background will partner with somebody who has more computing background. And so people can learn from each other that way. We've had quite a few experimental biologists who had no computing come into this class over the years, and certainly it's people who had strong computing backgrounds with no biology. Uh, and if you're coming from bioinformatics, it's a very different approach. We use a lot of bioinformatics when we build models, but the approaches are very different. And I think you could learn a lot from seeing these complementary points of view. Alex, I know you're an auditor, but maybe you should introduce yourself too.
So I'm a senior undergraduate majoring in biochemistry. I pretty much only just started this modeling stuff last semester, but before this, I have a fair amount of experience with in vitro modeling, in vivo, protein biochemistry. And I'd say my primary biological interest would be in cancer immunology.
So maybe combining that simulation of the tumor angiogenesis with the simulation of the viral infection response. Yeah, maybe we could take. Paul has a nice simplified model of immune cell response to a tumor. Maybe we could re-implement that in CompuCell. Take the PhysiCell model and re-implement it in CompuCell. It might be a good exercise. Okay. How shall we do this are you on the Zoom, Pedro?
No.
Okay, so for people who are online, maybe I should have people who are gonna in the room walk up and talk in front of the computer so that everybody can hear. That's okay.
Hello. So I am Pedro.
You talk to the audience.
Oh, okay.
But they'll just hear you if you talk.
All right. So I am Pedro. I have an undergrad in physics. I am studying bioengineering as a graduate student, and my research is on cell immune response to influenza A virus.
Hello, everyone. I'm Nishant. My undergraduate was in computer science, and my master's is also in computer science. And I took this course because I had an interest in biology in high school, and I figured combining bio and computer science was something interesting. And yeah, that's why I'm here.
You know, it may be worth saying something. I don't know if our online people can hear me from here, which is until I was a postdoc, I never had a course in biology. And the only course in biology I ever took was advanced neuroscience methodologies at Woods Hole. The experimental course in advanced neuroscience techniques. And so it's sort of an irony that I've spent 30 years of my life doing and teaching biology, both experimental and computational biology. And I never had even not one introductory course in biology. I had to learn it all myself. So I guess it means that I have certain gaps in my knowledge. I'll say that, but it also I think means that at some level it's interesting to come to a field from the side, because a lot of things that you see in the textbooks don't make sense. And if you learn it in the introductory biology course, you just hear it, this is how it is. And so people don't think about it. I talked about Bob Gatenby and his thoughts about cancer evolution at Moffitt. And for 35 years, people thought he was a crackpot, except he was right. Now, of course, you can have odd ideas that would be wrong. But in fact, he was a clinician. It wasn't that he was, but he was able to ask questions about, does the story about how cancer progression works make sense? And to him, the answer was no, that you had to think about it in a new way. And that led to very, very impressive advances in what are now thought of as immunotherapies and cancer therapies. And so sometimes I wouldn't say that it's easy. You have to learn biology. But coming to biology with an interest in it, but not as an insider, can sometimes give you new insights into how things work. You can ask questions about what makes sense and what doesn't. That work that I showed of Abbas's on the neoangiogenesis in age-related macular degeneration. The ophthalmological community was certain they knew how that disease worked, and they were wrong. And it had a big significance, because if you think that the disease works, now it's true, there are plenty of medicines that were developed before we knew anything about how diseases worked. So it's not that you have to know the mechanism for disease to be able to come up with a therapy.
Mm-hmm.
And even if you know the mechanism, you may not be able to come up with a therapy. But you're much more likely to be able to come up with a therapy if you have the mechanism. And so, and a lot, of course, of modern bioinformatics in terms of developing drug targets is about trying to do this. But there are a lot of factors in terms of some of these, what we call developmental diseases, which are about basically how processes in the body work. Where, as I say, looking inside the cell doesn't tell you what the problem is. And in those situations, this kind of analysis that we're doing here could give you real insights. It's a lot of work. It doesn't always work. It's not magic. I don't believe in magic. But it can give you really new insights into how biological processes work. And so I hope that people will stick around and explore this with us, because I think that there's a real richness, especially for people coming from, say, a bioinformatics background or from a computing background, in trying to find common ground together to understand these problems.
Okay, so my name is Wei Yan, but you can call me William or Will for nicknames. And I'm a junior in IC, concentrating in bioengineering.
One thing that I like very much about this class, besides the variety of backgrounds that people come to, is the fact that the graduate student, undergraduate, you're basically all on the same footing. Because, again, with a few exceptions, people generally don't have all the background. They haven't done this before.
Mm-hmm.
And so in that paper on COVID modeling that we published, several of the authors were undergraduates. And it was a relatively major paper. So it's no guarantee that in a semester you'll do publishable work, but it's possible.
My name is Emma. I am a second year PhD student in the cognitive science department with undergrad majors in cognitive science and math. I would say that I have a good amount of programming experience. Right now my research is on mathematical models of like the neural basis of navigation abilities. And for this class, I would be interested in focusing on the development of a particular cell type in a particular part of the brain. The cell type is called grid cells because they have these interesting grid-shaped firing fields. But if I get distracted enough, I might be interested in working on something having to do with prions or long COVID or something about muscle tissue because I think it's cool. So if you're interested in those things, I'm very distractible.
I hope that people will find projects that are of interest to them. I encourage people not to be too ambitious about what they try to do in one semester. Not because I don't think you should do big projects using these kinds of approaches, but just because, again, it's like learning a language. You want to do something that you want to have some small achievement under your belt first. And I will never tell people you can't do something, but I may encourage you to if I think something is risky, I may say so. But we'll see for that. Okay. All right. So let's actually now, we've run a Computational model in our last 20 minutes. Let's actually write one. Computational has a little tool called Wizard, which needs to be developed a lot more. And in fact, Pedro was working with some undergraduates last semester extending Wizard. But let's use the Computational editor now. And so I'd ask people to launch Twedit. Okay, so if people go back to their angiogenesis simulation, now I know there are people who like to use other editors, but for the beginners, we're going to use the Twedit. If you go to your angiogenesis simulation window and you hit this thing that has a picture of a piece of paper and a pen, it says Start Twedit here, or you could go under File and hit Start Twedit. It will launch the editor. Okay, now inside of Twedit, we will go to CC3D project, new CC3D project. And it'll pop up something that says Simulation Wizard. And here you can set the library where you're going to put your code. And I would recommend navigating to a directory where which you'll use for the whole class for your projects. So I'd recommend creating a directory somewhere in your writable user space where you can save your simulations. And I'm not going to tell you where that should be because it'll depend on how your computer is configured. Okay? So you could browse and navigate to a directory that you want. And we got to give the simulation a name. By default, it says new simulation. So we could call it, I don't know, my first simulation or something like that. The one thing that you want to be careful of, especially if you're on a Windows, on a Mac or a Linux machine, Windows is a little more forgiving. Don't put any spaces or special characters in your file names. And don't put a number at the beginning of a file name. So you can put numbers at the end of the file name, but don't number your simulation 1, please. Okay. So type the simulation name, hit next. And now you will have a window called Simulation Properties, which sets up the simulation. And by default, it offers you a 2-dimensional simulation, which is 256 by 256 lattice sites. And we are, for the purposes of this exercise, just going to accept those default values. And we'll come back and explain what they mean later. But for the moment, we'll just do default. So please say next. The next window allows us to specify the cell types. And we're going to create 2 cell types. We're going to create cell type 1, cell 1, and cell 2, and we could have those be bacteria and macrophages. There could be T cells and B cells. We could give them many names. At the moment, they're just placeholders for the fact that there are two kinds of cells. All right. We hit next. And the next thing that we're offered is chemical fields. We are not going to use chemical fields, so we're going to hit next. And then we are asked what are the basic cell properties and behaviors that we are going to use. And we are going to have the cells stick to each other, so we're going to hit contact. We're going to have the cells grow, so we're going to hit growth. We're going to have the cells divide, so we're going to hit mitosis. Okay. Everybody keeping up with that? We can walk through it again. We hit next. It will say configuration complete. Okay, so let's see. We do. Not sure what happened. Why it would die on me. Let me see if it actually created the file. Possible it created the file. How about Jonathan? What did you get? You want to screen share it?
Yeah, I'll pull it up.
Yeah, so for you, it looks like it's okay. Let's hit run. No, for you, it's also dying and disappearing. We forgot to click volume flex.
Okay, back in Twedit.
Go back to Twedit. Go to CC3D project, new project. Okay, great. Okay, hit next. Hit next. Okay, cell types, create two cell types. Okay, good. Next, next, skip. Yep, next. Okay, hit contact. Growth. Growth. Not secretion, growth. Hit growth. Mitosis. And then volume. Growth.
Local flux.
Yeah, that's right. Fine. Now hit next. And now hit finish. Okay, now run it. Okay, so you got it. Okay. So that's actually a simple avascular tumor simulation. So thank you very much for sharing. I want to talk a little bit about how simulations work before we break. There are two components to most simulations, a markup language component in CC3D ML, which specifies static properties of the simulation, and a Python component that specifies dynamics. In Twedit, if you double click on the file name, it'll open the windows and you'll have tabs. And one tab will be the XML, one tab will be something called Python main, which we won't use much, and one will be called Python steppables, which we'll use. In the simulation specification in the XML, you will see some metadata that says things like debug and parallelism, a section called POTS that says how long the simulation runs and how big the simulation is, a section that will tell you what the cell types are that you declared, a section that will tell you how adhesive the cells are to each other, and a section that says what the initial conditions are. In this case, we drew a square box with two kinds of cells in it. If we switch to the Python, we will see a lot of Python boilerplate, and we don't have to worry about the Python boilerplate. That's provided for you. And we will see three functions inside of the Python. A start function, which is run at the beginning of the simulation that initializes things. And in this case, we will see for-cell and self.cell list, which we'll use a lot. That does something to every cell in the simulation. And in this case, we're going to set the volume of the cell. And the reason the cells were disappearing was we forgot to invoke that function to set the initial volumes of the cells. We then have something called the step function. And the step function is called typically every time step, although you could adjust how frequently you call it. And in this case, in the step function, we go through every cell and we adjust the volume of the cell. And in this case, you'll see we're adjusting the target volume of the cell rather than the actual volume of the cell. And next time we will talk about why that is. Why you have to adjust the target volume, not actual volume of the cell. But fundamentally, the cells are soft objects. and they adjust to their environment. So we can't force them. Suppose we had 100 balloons and we tried to stuff them into a small box. We can't specify the volume of each balloon because they can only fill the space that's available to them. And so in this case, we're basically creating balloons, which are cells, which have a volume that they would have if they're not compressed. But if we compress them, they'll be smaller. And if we pull on them, they'll be bigger. And so we have the cells grow. Then we have a cell division algorithm, which creates an empty list. So that's our first real Python. Cells to divide equals empty list. We say if the volume of the cell is bigger than 50, we put it on the list to divide. We then run through the list of cells to divide and divide them. And we have a variety of ways we can divide them. We can divide them randomly, we can divide them in an oriented way, and so on. So we have self-divide cell, which divides the cell. Once the cell has divided, we now have a new cell. And that new cell by default doesn't have any attributes. And so we use a function which is called clone parent to child that says that the daughter cell has the same type as the parent. If we did that by itself, the daughter cells would also, both the daughter cell and the parent cell would have the same volume, so we would have doubled the volume. So we first, before we copy the cell, we say that half the volume goes to one cell and half to the other. Self.parent cell target volume over 2. And then to make things a little bit more interesting, we swap the types of the cells. So if the cell was a type 1 before, we make it 2, and if type 2, type 1. And that's all that we have in that simulation. So it's actually, it has a little bit of Python, but it's really not that bad in terms of code. And it's a relatively sophisticated simulation in the sense that you have cells that are moving around, they're growing, they're dividing, they're changing type. And so I appreciate everybody's patience today with the problems with the simulations and getting things up and running. We will check to see what's going on with Wizard on Windows.